T-minus 1 minute. T-minus 30 seconds. We are go for launch. T-minus 10, 9, 8, 7, 6, 5, 4, 3, 2, 1.
Everyone, today we're at Kennedy Space Center at our Space Station Processing Facility, and we are going to give you a tour of everything that goes on here. So follow me. We're on our way to our ISS Environmental Simulator Chamber area, which is one here on the ground. So our scientists can do investigations in these chambers while doing investigations on Space Station, and the only difference is gravity. On Space Station they have microgravity, and here we have Earth gravity. So here's our chamber area, and we have a number of large chambers as well as 2 smaller chambers where we're able to do these replication experiments. Some of the things that are going on in here are our verification tests before science verification test, which is really seeing how science works in NASA's hardware. Then we do an experiment verification test where investigators do a full-up dry run of their space station experiment, but on the ground. And finally, we do the ground controls for experiments that are running on Space Station. And at any given time, we'll have different hardware and different types of experiments going on in this facility. One of the things that we have in here are some veggie chambers, and you can see the veggie chambers that are being set up to run a verification test on an advanced plant experiment, or an APEX experiment, which uses Petri dishes. So that's why we have those racks in the chambers. We're going to go down the hall now to what we affectionately call our dirty lab. This laboratory is also special because we have photographs of all our previous interns with their signatures on the door. So here is a place that they could really show that their impact at KSC from their time. So we use a lot of different substrates in our experiments. You'll see turfus, you'll see vermiculite, and even a potting mix, because we'll use that sometimes as well as perlite, coconut coir, which is a fibrous media that we sometimes use. And I think so, depending on what we're studying, we may use different substrates or media for different purposes. Where we can dry plant tissues, we bag things up and oven dry them to get dried weight or to do certain types of chemistry. This space is designed to be able to be reconfigured depending on the needs of a particular investigation. So So now we're showing the doors to our main plant processing area, spacecraft production area. The first door we have here, where we have the seeds and the advanced plant habitat laboratory and a lot of equipment storage, is our Earth door. And we talk about how NASA plant research has increased crop yields on Earth and enabled solutions for space exploration. And you can see the rocket launching from the end of that cornucopia there. We go from Earth to the International Space Station, and you can see Veggie up there, and we talk about how we're studying optimum growing conditions for plants and crops in space aboard the International Space Station to enable future exploration. And this is the door for our Greenworks laboratory. We have a smaller human-sized door we enter with some aliens on it in a future crop production system. And then the doors for our main plant processing area space, where we have the chambers and where we're going to go live, are the moon. And here we have a lunar surface greenhouse depicted. And we talk about how fresh food in space will be an essential part to the solution of the challenges of long-duration human exploration of the moon and beyond. And we have another entrance to the same room showing Mars and what a 3D printed Mars habitat may look like, or a Mars plant growth chamber that could be sent on a module to Mars. And we talk about how solving the challenges of the harsh environment of Mars will lead to new solutions to feed people on Earth.
Hi, everyone, and welcome to today's amazing live stream. My name is Samuel Garcia, Education Specialist at NASA's Kennedy Space Center, and we are standing inside the spacecraft production facility at NASA's Kennedy Space Center. And with me is this wonderful, amazing team of scientists and engineers, which we're going to hear from in just a moment. But before I do, I want to invite each and every one of you to tell us a little bit about where you're joining us from in the chat. We want to hear from you, so make sure you let us know where you're joining us from, because you are those amazing students and schools that are carrying this important work forward. So thank you for this amazing work that you are doing. So I'm going to turn it over to our first guest. If you can tell us a little bit about who you are, sir.
Hey, I'm Trent Smith, and I work in the Technology Transfer Office here at Kennedy Space Center, and we make the inventions that are made here and at other research facilities at Kennedy Space Center available to industry.
My name is Christopher Bermudez, and I'm a spacecraft production intern here at Kennedy Space Center.
I'm Joya Massa, and I'm a plant scientist here working in spacecraft production.
My name is Jacob Torres, and I'm a plant science engineer in the plant lab.
Hi, I'm Christina Johnson, and I'm a NASA postdoctoral fellow here at Kennedy Space Center working with these folks.
All right, team, thank you very much for those awesome introductions. Now we're going to kick it over to the GBE team in Miami. Amy?
Hi, everybody. Hi. Welcome, and thank you for joining us today. We're so excited to be collaborating with this amazing team. I'm Amy Padoff, the very, very proud Director of Education at Fairchild Tropical Botanic Garden. And since 2016, Fairchild has been partnering with the scientists at NASA's Kennedy Space Center to advance the research on plant science. And since the program's inception, students' findings have influenced the plant selection and harvesting techniques implemented in NASA's Veggie production system aboard the International Space Station. We have, by participating in this great program, Growing Beyond Earth, more than 40,000 students in 48 states and 6 countries across the world have been collecting and contributing important data to NASA plant science, working directly with the plant scientists, this amazing team at Kennedy Space Center, developing and conducting original experiments, analyzing the data and communicating the results, and collaborating with students nationwide. I want to send out a big thank you to those of you who are working on their first trials this year, and those of you who are not, we're excited to announce a new mid-year training for new participants beginning in January. So it's a new cohort coming on. We're very excited. If you are not participating and you're interested, or you know somebody who might be, please check it out on our website at www.fairchildgarden.org/GBE and sign up for the upcoming workshops. Also, if you haven't, check us out on Twitter at Grow Beyond Earth and follow the students as they conduct their experiments. Once again, thank you so much for joining us today. We're so excited to have you here. Thanks so much. Back over to you.
Much GBE team, Amy, for that wonderful welcome. So now we are here with Trent. Trent, can you tell us a little bit about what we see here in front of us, this whiteboard with all this different text and different, I guess, projects that you have here?
Yeah, you bet. We sort of refer to this as Ralph's board. Ralph Ritchie's our senior project manager for space crop production, and he drew this all up because there's a lot of questions we don't know. You know, for instance, the end game is to be on Mars and growing fresh, nutritious food for the astronauts on Mars and for the return back. But the question is, how do you do that? So there's a lot of questions we need to answer, and it all starts with ground research here at Kennedy Space Center. There's thousands and thousands of crops. How do you select them? These crops have to grow in a space vehicle or on a lunar base or a Martian base. We expect those to have high CO2. Going to be growing under electric lights, probably LEDs, and you're going to have partial gravity. In some cases, you won't have a magnetic field. So all these changes are different. Every plant that you've ever heard of or ever grown has all evolved under broad spectrum light with, atmospheric pressure and magnetic field in 1G. You change these things and the plants know something's different. So this is why we do research on the space station. So there's microgravity, high CO2 and LEDs. So we're just learning how to grow and eat our food on space station, and all of that comes out of the ground research. So as you move out away from low Earth orbit, now you have microgravity and radiation, right? So how will radiation impact the seeds and the plants? Right now, radiation is one of our biggest risks for the astronauts. And as plant scientists, we're worried about the plants too, right? So we want to sort of understand how the radiation might play a role with the plant growth on the gateway and microgravity, and then on the lunar surface where you have 1.6G, right? So these things could impact you. And then on the way to Mars, what are you going to be doing for that long transit, right? It's a long ways to Mars. And so one of the things that folks are thinking about is to have astronauts grow and eat food, because it can provide that connection back to Earth. You know, the astronauts go around the Earth every 90 minutes roughly, and they can see the Earth, but they can't touch it. So when you're growing plants on the space station, it can give an astronaut the connection back to Earth, the smells, the touch, and the feel of back home. It's a very artificial, extreme environment, and having something growing and living along with them can provide a lot of boost. So, you know, now you take that and say, now that blue marble that's getting smaller and smaller and smaller, and you're on your way to Mars, and anything that you've ever heard of or anything that you've ever known happened there, and there's no connection. So maybe those plants can be a really, really big, boost for the morale and for psychological health. So again, on the way to Mars, you're going to have microgravity and radiation. You're going to hopefully be growing some plants for nutrition. And then certainly on the Martian surface and on the way back, you want to be sure you have vitamin K, vitamin C, and B1. Now, plants are molecular architects. They can provide these if you grow them and eat them. But it all starts here with ground research, and it all starts with citizen scientists running experiments for us and providing the data in the Google Sheets. We've already flown Fairchild crops on space station. We had dragoon lettuce, we had extra pak choi, and it all started with research done with the citizen scientists. And so all of that feeds to space station, to the lunar, the Artemis, right, our Artemis program, to the lunar gateway and the surface, and on the way to Mars. So how will you grow and eat your food on Mars? It all starts right here with what we're doing. And the citizen scientists running the experiments, where the answer is not in the back of the book. We get the data and we learn.
Trent, thank you very much for sharing that. And students, you have a task, right? You are doing this important work. So your challenge is to continue supporting this amazing research at the ground level so one day we can send humans to Mars. Now we're going to kick it off to Chris Bermudez, who was a former GBE student and who is now an intern here at NASA Kennedy. Chris?
Hi, my name is Christopher Bermudez, and I'm currently an intern here at Kennedy Space Center's plant production area. I first started in biotech at Richmond Heights. It was a magnet high school which had a botany magnet, and that's where I first got my passion for, well, the research we do here. I initially began with the Growing Beyond Earth program, going to Fairchild Tropical Botanical Garden. So now that I'm in college, I was able to intern here. As with the Growing Beyond Earth program, we're given all the tools and materials we need to do, and we just follow through in growing the plants. But here we need to be a little bit more proactive and think outside of the box when it comes to these experiments, as these are flight experiments that will one day go to the International Space Station. Not a lot of people believe that being a farmer is a valid career path, but what we're pretty much doing is space farming. So I believe that a lot more students should get involved in projects like this. Best of luck to the rest of you, whoever decide to continue following within the pipeline that is Growing Beyond Earth or even NASA internships. Because the more you apply yourself to the program, you can maybe one day end up like me, working here within the plant production area at Kennedy Space Center.
Thank you very much, Chris. Now we are here with Dr. Christina Johnson, who is a postdoctoral researcher here at NASA Kennedy Space Center, who's going to tell us a little bit about her research around microgreens. Dr. Johnson, thank you very much for joining us.
So I work with microgreens. Microgreens are plants that are at the very early stage of development, between germination and about 2 weeks old is when I'm looking at these plants. And I really like working with this stage of development, because if we get it really good, if we get everything optimized at this stage, then they're just set up for great for the rest of their life. But also, we can eat them at this stage. And I love eating them at this stage because they are packed full of nutrition. If you have a handful of microgreens and a head full of cabbage, they actually have very comparable nutritional contents. And so would you rather take all that time growing a whole head of cabbage and then eating a whole head of cabbage in one sitting, or would you rather enjoy a little hand salad of microgreens? I'm kind of partial to the hand salad of microgreens, especially since you can have a big range of flavors and colors and textures with those microgreens. So today here, I'm growing radish microgreens, and I'm growing them a few different ways. I'm growing them in simulated microgravity, and I'm also growing them in just an elevated carbon dioxide environment like what we'd find on the space station. Can I pull out a tray for you and show you what I have here?
I think the students would enjoy that. Yes, please do so.
Young plants. I'm actually going to harvest these in a couple of days. So they're really leafy and little, and they have red and green colors to them at this point. And so this is what they look like when they're just growing in a tray. And this is kind of what you'd see for commercial production of microgreens. If you were to get them at a restaurant, this is how they would be grown. And in comparison with what I'm doing to get them ready for space flight, I'm running a bunch of simulated microgravity runs where I've got them in smaller trays growing on these special random positioning machines. And I'm looking at their development, and I'm making sure that they are able to have adequate nutrient delivery, make sure that they've got enough water to them, and also making sure that they're really hitting those developmental milestones that we're expecting out of them within a time frame that we'd expect. And same kind of microgreens are growing over here I have some stationary controls, and I have some that are constantly moving. These ones are older than the ones down there. The ones down there, I have these cute little covers. So I put a cover on it to make sure that I have enough moisture trapped in there so that they have a high moisture environment during germination while they're just coming out of their seed coats. And then I take this off after a couple of days and I let them grow with the lighting and all of the other aspects. So it's, yeah, these are 3D printed. A lot of this, to enable this, this working with the simulated microgravity environment, really revolves around 3D printing. And I'm very dependent on the engineers. I'm a plant scientist. And while I love engineering concepts and looking at engineering tools, I don't have those skills. And so I depend on people like Jacob Torres to make this all happen. And I love being a part of this really interdisciplinary team to make all this work.
That's wonderful. Christina, thank you very much for sharing a little bit about this amazing research. And speaking of engineering, we will hear from plant scientist Jacob Torres around the intersection between plant science or space crop production and engineering. Jacob?
Hey everybody, my name is Jacob Torres and I'm a plant science engineer at NASA Kennedy Space Center, where I get to grow plants in space. Building hardware and designing equipment that makes plants grow and plant experiments grow on the International Space Station. Behind me is one of my printers that's getting ready to start printing. It heats high enough to print flight-approved parts or flight-ready parts. So this 3D printer is a regular 3D printer or a low-temperature 3D printer that you would see in most labs, but it has limitations in temperature to print different materials. This printer goes up to 400 degrees C and can print flight-ready parts. And what we're seeing here is actually one of the hardest materials that I've ever printed before. Is called polypropylene. It doesn't stick very well to surfaces, and we're printing out the test pucks for the biofilm formation testing. These are different types of materials that are available that we can use to make hardware out of. And understanding how these materials behave and what they can do and what you can use them for is really important for our research. So I'm printing them out here, and we're doing different tests where we're growing plants, doing germination studies, doing tensile and hardness, flexural testing with them, and then we send them over to the microbiology group who's going to do biofilm formation testing on them. For example, Plant Habo 4, PH04 grew the first fruit in space. It was a pepper called the Munex Espanola Improved, and it needed hardware to help it to grow. So the Advanced Plant Habitat has a box that contains the growth media and where the roots grow into, called the science carrier. The science carrier needed specific wicks to allow the peppers to grow, and my first 3D printing job was PH04, where I 3D printed these wick pegs. And all they did was hold the wicks open to draw the salts away from the stem, and that way it helps the plants to be more successful. The salt would eat away at the stem and you'd see stem rot. Let's see some 3D printed parts in action. We're getting ready for our next spaceflight experiment called PH07, Plant Habo 7, and we'll grow lettuce on the International Space Station to test the microbiome of the root zone. Here are some 3D printed parts that are making that experiment happen. Inside you can see the growth boxes that the lettuces are growing in. Those parts are 3D printed. I get to have a lot of fun taking the parts from the makerspace and putting them to work in the plant lab, and eventually they go to space. All of this work is the stuff that I love to do the most. I do this at home as a hobby, and I get to do it at work as well. And we have so much fun doing and building space biology hardware.
Awesome. Thank you very much, Jacob. And we are standing here with the real Jacob Torres. But first, we want to give you and remind you, students, please make sure to write down your questions in the chat, because we want to pull those at the end and have this awesome team of scientists and engineers to answer your questions. So this is your chance. Don't miss out to type in your questions in the chat. So we have Jacob Torres. He is a plant science engineer. And just recently, we heard from Dr. Johnson about ground-based microgreen research. But just recently, those microgreens were taken up on a parabolic flight experiment. And the team here wanted to learn more about how they cut and harvest in a micro-simulated environment. And Jacob was on that flight. Can you tell us about that experience, Jacob?
Absolutely, Samuel. I was so honored to not only be on the parabolic flight where we tested different harvesting methods for microgreens, but I also built hardware that flew on that flight, which was really cool. And we ended up going in a glove box that was a lot like this. We went down to Fort Lauderdale Airport where we got on a zero-G flight. We created some different plant growth methods to grow microgreens, because like what Christina Johnson was showing, we want to grow crops, microgreens in space. They're a good short-term crop with a lot of nutrients, but how do you do things like planting them? What type of hardware do they need to grow? How do you harvest them in microgravity? How do you put them in the bag for astronauts to eat them? Those are all things that we have to learn, and we use the parabolic flight as an opportunity to learn that. So we had a glove box just like this that we borrowed from the University of Louisville, and we set up our plant growth demonstrations inside. We actually started growing microgreens in our hotel room before we went for our flight. So we had about 53 boxes just like this one, or another one like this. We had two different versions, two different manufacturing methods that grew them. And what we wanted to do was try to see different versions or different ways to grow microgreens, see and understand what grows best, what doesn't. In this case, it was an all 3D printed box. It actually has a reservoir in the middle. It has a growth space in the top, and then it has a cutter on the top part. So it's made to fit together and contain all the moisture and the media and the seeds and everything in one spot. You add water, grow microgreens. The microgreens actually grow through these holes here. So you have to place a seed in each hole and allow them to grow. And about 8 to 10 days later, they're ready to harvest. But when you're in microgravity, how do you harvest them without them floating everywhere? So one method was to do what we call the guillotine method. And this one has a little blade that you slide down. It cuts the microgreens, and you can grab them right here and add them into your bag. So it's all 3D printed. But we also have a prototype shop here at Kennedy Space Center. That's a fully equipped shop that you can build and cut equipment and materials. And this one was made by Larry Koss. And his idea was to do a salt grinder type of method. And the salt grinder where you would grow the microgreens in each hole, place a seed in each one, grow the plants. And then once they're mature enough, you turn them and it cuts the plants off. In microgravity, that would release the plants up. And so how do you harvest those bags, and how do you harvest those plants, and how do you keep control of them? That's where the bags come in. So we tested with many different bags that would fit around the hardware, and they're all designed to capture the microgreens and really trying to find a way to capture them easily, but also give the crew easy access to eat them. So we had different versions of bags that fit on just like this. When we did the guillotine, sometimes you can cut the guillotine and hold the plants all right here. We also did one where we cut them with scissors, and maybe that didn't work as well, but some of those results we'll see in a video coming up, and it's really, really exciting. But the prototyping, the maker area, rapid prototyping directly supports experiments and plant growth that are going on in space, but also here in the plant lab. And we get to build a lot of things like Christina Johnson's UGrow. She was talking about growing microgreens on a random position machine or in simulated microgravity. How can you grow plants? And when you're growing microgreens in a microgreen environment, well, there is no top, there is no bottom. So why not grow on both sides? So in this case, we created a 3D printed box that has two growth surfaces. You can grow on the top and you can grow on the bottom. Fill this up with nutrient solution, put the wick on top, place seeds, and you can grow the plants that Christina was showing just there. So all of these things are 3D printed, rapid prototyping, maker things that we build here in the lab, and then they're applied for different experiments. And it's so much fun because that's what I do at home for fun, right? I have a 3D printer at home. I do CAD animation. I usually do flowers and other things, but this time we're making space biology equipment. So the things that you're doing at home, your hobbies and the skills that you have a good time doing, have relevance to space biology and other NASA missions as well. So all these things that you're learning and you're doing in school are going to contribute towards who you're going to be in the future. And that's a really good way. And in the end, you know, we're going to have many missions coming up, missions to grow in microgravity, missions to grow plants on the moon, later on Mars, and we will need more space farmers and more helpers to help us do that. The engineers and the plant scientists of the future are all of you guys, and we're excited to have you. So with that, I believe we're going to take a look at the parabolic flight results, as well as introduce the advanced imaging team to take a look at some other things.
All right. Thank you very much, Jacob. That was awesome. So yes, let's cut to those videos so the students can see exactly what Jacob was talking about. And that video was cut by our very own GBE student, Chris Bermudez. So Chris, thank you very much for editing and creating that wonderful video.
Hi, welcome to the plant processing area. I'm Aubrey O'Rourke. I'm a project scientist at Kennedy Space Center, and we're going to show you the hyperspectral imaging project that we're working on. Come on over. All right, so here we're growing Dragoon lettuce. We have a treatment with a moisture, which we are surveying here with moisture sensors. We have our watering lines feeding the plants to control how much water they're getting. We can see our imager has begun to take the light panel reference for reflectance, and we'll begin to scan over the banks one by one over the trays to take wavelengths of reflectance to assess crop health. So in spaceflight, we often come up with the concern of how much we're watering or underwatering the crops, because it is hard to water in spaceflight, particularly in microgravity. Oftentimes water in microgravity will cling to the surface and essentially starve the roots for oxygen. In this case, we want to be able to monitor that phenotype before it starts to have bad effects on the plants themselves. So here we control the watering. We take imaging of these different treatments so that we can assess whether the plant visually or spectrally with our hyperspectral imaging is having stress response. The complete system will combine hyperspectral imaging with LiDAR, which is imaging from the top where the camera is to the top of the canopy to tell us exactly where we're seeing this stress. And we'll also combine this with thermal imaging for a whole multimodal imaging remote sensing capability that hopefully will fly one day on board our exploration systems. My background is in microbiology and chemistry, so the coupling of this research with the profile of metabolites in plant nutrients can make for a robust assessment of what do we see through imaging and how healthy are crops for our crew. this work is done in part with the USDA collaborators, and their work prior to this works with remote imaging of large agricultural fields, particularly using fluorescence imaging systems. Okay, this is the view from what is captured by the imager. It's taking line scans, and it patches together these line scans, each line scans being hundreds of wavelengths, so we're capturing lots and lots of data here. These images will then be processed by our collaborators at the USDA, and from that we'll get information about which reflectances are most indicative of our stress that we're surveying. These stresses can be water stress, they can be nutrient stress, they can be salt stress, and the objective is to detect the stress before it becomes a problem for the plant production and before the human eye can detect this stress as well.
So now we are here with Dr. Joya Massa, who's going to tell us a little bit more about what other things and what other areas of research they're currently conducting in this lab.
Welcome, students. We're really excited to have you with us today. Growing Beyond Earth is a huge part of our program, and we want you to know how much you're contributing. So follow me to see some more of our lab. Here we've got some etc. chambers. We call them etc. chambers. It stands for environmental test chambers. And these are smaller chambers, which are actually the size inside of our advanced plant habitat on the International Space Station. So advanced plant habitat, as you may or may not know, is Veggie's big sister. It's a controlled chamber that has lots of different sensors and monitors and controls every aspect of the environment. These etc. chambers allow us to do independent research studies in smaller chambers, and we can put a whole bunch of them in one of our larger chambers that you've seen before. And we can look at temperature, carbon dioxide, and humidity and control them separately in these chambers. These particular chambers have actually been built by Larry Koss, our mechanical engineering designer, for Epcot Center, and they're going to be going on display in Epcot Center once we get all the bugs worked out of them. So they're a really cool system to control the environment for crops. And we've got some outrageous red romaine lettuce growing in this one. These are some of our small chambers. They're called reach-in chambers, unlike the larger one you saw Christina in, which is a walk-in chamber, or you saw Aubrey or Jacob in the walk-ins. These are smaller reach-ins, but they do the same thing. They control our temperature, our relative humidity, and our carbon dioxide, so we can set the conditions to mimic what we experience on the International Space Station, so that the plants growing inside kind of think they're in space, except with gravity. What we're doing in this particular chamber is part of our new crop testing and our crop selection, which is what Growing Beyond Earth has really contributed to over the years. And on the top shelf here, we have strawberry plants. Strawberries are a plant type that we've been looking at, and we're looking at different varieties of strawberries that actually grow from seed. So if you've grown strawberries in the past, you may have noticed that most times they come from cuttings, from little root cuttings that you buy and then you plant. Very few strawberry varieties actually grow from seed, but there are a few, and we've been testing those to see how suitable they are for spaceflight. And so strawberries take a long time to grow, especially from seed. And then they start sending out these runners. You can see some of those red stems there. Those are above ground stems called runners, which will make other little plants along the way, little plantlets. And then eventually they will start flowering and fruiting. These plants are a little too young, and they're not at the point where they're flowering and fruiting. when we've done some other strawberries, we actually were able to grow the same plants for over a year and collect fruit for most of that time. And we would have to go in and pollinate the flowers, just like the astronauts will have to do on space station if they're growing, fruiting crops. At the bottom, we have our red robin tomatoes. These are the tomatoes that we are going to be launching to the International Space Station as part of our VEG-05 mission, which is going up really soon. November 18th is our launch date. So we're very excited to grow tomatoes finally in space. This red robin variety was selected from a whole bunch of different tomato types that we grew in these chambers here, and we screened the different crops. Now, Growing Beyond Earth hasn't worked with strawberries or tomatoes yet because they just take a long time. Tomatoes take about 80 days to start fruiting and so that you can pick the fruit. And they'll fruit for, over 100 days or so. Strawberries take about 100 days just to even start flowering and fruiting. And then, you can continue to harvest strawberries for a long time. But those times don't work very well with the school year. So that's why Growing Beyond Earth is testing leafy greens, herbs, and last year radishes. Though we did do chili peppers a couple of years ago, and Jacob helped lead that research, and we got a lot of really useful information from the chili peppers, but that was when we were all working from home. So we do a lot of different types of crop selection here. To figure out which varieties are going to be suitable for space and which will grow well in our environment, which will be very productive, which will taste good for the astronauts. We want delicious crops, and which ones will be very nutritious. We also look at microbial food safety for our crops. So all the experiments that you're doing in your classrooms help to feed into these types of tests that are going on here at Kennedy Space Center. And ultimately, they'll help to feed into the tests that get tested on the space station and someday on the moon and onto Mars. Thank you all for your work with Growing Beyond Earth. We really can't do this without you.
Awesome. Dr. Massa, thank you very much. And I think it's time for the questions, if I'm not mistaken. And yes, we're going to go ahead, kick it back to GBE right now, and we will see you shortly.
Yeah, great. Thank you so much. Thank you all. I'm Carl Lewis. I'm the director of Fairchild Tropical Botanic Garden, and I'm part of the GBE team. I'd like to thank you all for joining us. You've already heard from Amy Padolf, our director of education, and Marian Litzinger is here with us today. She's the Growing Beyond Earth project manager, and all of you who are participating in GBE already know Marian. She organizes this whole big citizen science project. And a very special thanks to the team at Kennedy Space Center for showing us their incredible work and how your classroom research fits into everything that they're doing. I'd like to give you all a sense of what it means for us to be part of such a great community of citizen science researchers. As a botanic garden, we study the history of plants and botany, how plants came to exist, how they sustain life, and how plants have been involved in the expansion of human civilization on Earth. And we also do things like restoring threatened and disrupted natural ecosystems. We work to return those ecosystems back to the way they were in the past. But we are also interested in looking to the future, thinking about how plants will support travel beyond Earth orbit, this next big leap for human exploration. And all this work is also going to shape the future of agriculture and how we use technology to improve food production here on Earth. So we love the fact that Growing Beyond Earth lets us explore the past, present, and the future of botany. And for the citizen science part of all this, we're interested in building as big a team as we possibly can with as many classrooms and students as possible. We'd like to put all of this research, which is so important for our future, into the hands of the students who are going to see all of this come to fruition in the years and the decades ahead. And it's for us, it's inspirational to see how passionate the students and teachers in GBE are, and how many great new creative ideas come back to us as a result of the work that goes on in all the classrooms. So to everyone participating in Growing Beyond Earth, thank you so much for all your hard work and your creativity. We hope you're having as much fun with all this as we are here, and we can't wait to see the results of your research. And for anyone who's not already participating in Growing Beyond Earth, you can sign up on behalf of your school at fairchildgarden.org/GBE. We're constantly recruiting new schools. We'd love to make this team even bigger. So from all of us at Fairchild, thank you all so much.
All right, thank you very much, GBE team, for that awesome piece of information. Now we're back with the whole team here, and we're ready for some questions. So I think we'll start off with the first question, Jacob.
What percentage of astronauts' daily diet might be supplied from space plants?
That's a joy question.
You know, it's going to depend on the plants. So right now we're really focused on the fresh pick and eat crops. And our goal would be to get maybe 5% of the diet with these. Most of these pick and eat crops don't have a lot of calories, right? That's why they say eat salad. It's not the salad that has calories, it's the salad dressing. So as long as they're eating a lot of salad, it's really good for them. And it may not make up a large percentage of the diet by calories, but it could be a big part of their diet every day. As we go farther and stay longer, when we're, say, on the surface of the moon or on the surface of Mars, we may have the ability to cook. And then we can grow some additional crops like potato and sweet potato, wheat, rice, soybean, peanuts, dried beans. All of those can contribute to the space diet in the future. They require more processing equipment, but they, you know, ultimately we could grow the entire vegetarian diet if we had enough space. So we're going to increase the amount over time, but for now it'll mostly be salads and some herbs.
Hopefully strawberries.
And hopefully strawberries, yeah.
That's a great answer, Joy. I have a question that maybe Christina Johnson may be able to answer. Seul Shaya, I may say your name wrong. She wants to know what fabric you used in the U-Grow and why.
So that's a great question. I chose a bamboo felt material that's a commercial microgreen growing product, and it is really great at staying clean. I can autoclave it before I use it. A lot of materials, they won't wet up right if you autoclave them. Autoclaving gets them really hot and really puts a lot of pressure on the material. So if we can have a material that survives that process, it's really a wonderful thing. So it wets right back up after I autoclave it. I get the plants to grow on it. It stays pretty clean throughout the grow out, and the roots grow into it as well as on top of it, and it makes for a really good microgreen substrate. We've tried a lot of different things, but that's my favorite.
Yeah, maybe in the future you might be able to reuse it, right? When you start doing crop production.
Thank you, Christina. That was awesome. So we have another question from a friend of ours named Lori Waters. Lori has a question, a two-part question. Will there be a veggie and an APH on the Gateway Moon Station? Also, will Artemis include a lunar greenhouse as part of the surface habitat?
I think Gateway's really going to be a small research facility, so I don't know what plant science is going to be accomplished there and what volume, right? So I don't think it'll be a big plant growth system like APH or veggie, but
something for microgreens if we do, you know.
right, maybe something for microgreens or something for fundamental spaceplant biology. But you know, we always dream big and hope. We can always hope. Certainly on the lunar surface, we're just in the planning parts of that, right? Where we're just getting power stations planned and put out for bid so we can build them and have them landed. So that's all in development right now. But the hope is that we will have those types of facilities on the lunar surface.
Yeah, there have been a couple of calls for commercial lunar payloads, and I know a number of plant folks, including our group, have actually put in proposals. So none of these have been selected yet, but stand by. Maybe we'll actually get to grow some microgreens or other crops on the moon. That would be really cool. Eventually, though, I think there'll be plenty of plants growing on the moon because the astronauts really want them.
They're hungry.
Yeah.
Moon peppers. That would be a good thing. Awesome. Thank you, guys. Lunar chili. Let's see. We have another question from Megan Kenson. How do plants respond when they come back to Earth? We may not.
When they come back, they're typically frozen, so there's no response. That's an easy question. I don't think we've brought any live plants back that I'm aware of. Most of the time, we'll harvest them and then we'll freeze them at -80, so we preserve all the chemistry, and that way we can analyze how did they respond in microgravity, including the microbiology. So yeah, no live plants brought back that I know of. So we did have zinnia seeds. Thanks, Christina. We did have zinnia seeds come back as flowers, and we tried for a long time to get them to germinate, with no luck until was it Lane did, right? Lane, she had the magic touch, and we had some space flowers, second generation that we grew, and they look pretty good. Maybe some mutations, but mostly look pretty good. So I know we've had tomatoes go to space and a few others, but for the most part. You know, we don't know, or there's been very short microgravity experiments with plants, but they know they're in microgravity.
That sounds like a great experiment to do at some point. All right.
Some chili peppers back, right?
We did.
Time to test them?
We actually brought peppers back. We didn't get to grow the seeds, but we did nutrient analysis and elemental analysis on the peppers that came back. So that was really exciting.
Yeah.
Really cool. Let's see. I have another question from Kelly Taylor. Kelly Taylor asked, is it possible to get the drawings from the 3D printed parts? And the answer is yes, in some cases. In fact, there are drawings that are available from the Microgravity Simulation Support Facility. We've made different fixtures that fit onto the random position machine, including Christina Johnson's UGRO, which is also included on there. So there are parts that come out through the technology office where we allow the drawings for individuals to 3D print or print in your classroom and other things. But not all of them are available just yet.
But the 3D print files are available on the NASA technical report server, right? So we could maybe find the I could see if we can't provide the link to that report. Yeah. We wanted to be sure that was available. Yep.
All right. I think we got one last question by our friend Susan Jeck. She was wondering how much water needs to be generated each day to support plant growth for long-term space flights.
Guess that depends on how many plants you're growing, right? So that's a Joya question.
Yeah, it depends on how many plants you're growing and what you're growing them in. So, you know, we talked about veggie, we've talked about advanced plant habitat. There's a really big difference between those in that one is open and one is closed. So veggie is open. It uses the space station environment to grow the plants, including the humidity in the air, which means that as the plants transpire and lose water through their leaves, they're putting more water vapor into the air that's going back to the space station. The advanced plant habitat is considered a closed system, so it can recondense the water out of the air and reuse it to grow the plants. So in advanced plant habitat, we really start with a pretty small amount of water and we reuse it over and over and over again, whereas veggie's doing that, but on the scale of the space station. The space station collects that water, condenses it, and reuses it. So while it may take around 60 to 70 liters of water to grow tomato plants all the way through in 2 veggie units, most of that water, 90 or 99%, is going to be condensed and reused on the space station, where they recycle almost all of their water. Very little is trapped in the plant. And if you think about how much a plant weighs, 90% of that is water. So if you weigh your lettuce plant or your tomatoes or whatever, 90% of that weight is water. That's what's locked up in the plant. The rest of it that you add goes back into the air.
I've got through all my questions.
So did you want to mention microgravity simulation?
Yeah, so one question that came up was, how do you simulate microgravity here on Earth? And there are a few different ways that we can do it. And what it really comes down to is, for plants, we just confuse them. we're constantly moving them. We're constantly changing their direction or just moving in one direction constantly, depending on which instrument you're using. With the ones I have there in the chamber, I'm using it, a random positioning machine where it's constantly changing, constantly moving, rotating, goes in another direction, shifts modes, goes in another direction, and it'll shift speeds as well as the directionality. And so it makes it so that those plants can't sense the gravity. Plants have sedimenting starch grains that kind of move to the bottom of the plant cells, and that helps them to sense gravity. That's one of their main gravity sensing mechanisms. And they can't sediment, they can't move to the bottom of the cell and signal that gravity vector if they're constantly changing and moving directions.
Confused plants.
All right, so I think before we close, I just want to thank all the students out there. Thank you very much from the team here at Kennedy Space Center. You're awesome. You're doing amazing work, and we look forward to seeing you one day here working in the labs with this amazing team. So continue. Thank you, educators, for your support as well. And we hope to see you soon here again one day, working here with us at Kennedy Space Center.
And we look forward to seeing your data.
That's right. So continue collecting that important data for us. Thank you.